Welcome to Monobiology. In this lesson, we're going to learn more than you ever wanted to know about the cell membrane. As you can see from the picture in front of us here, the cell membrane is actually quite a bit more complex than you probably would have noticed from the other drawings and the other diagrams that we've been using. And the cell membrane is a very, very important piece, a very important part of the cell for lots of different reasons, but it's also a very important part of this course because as we move forward, we're going to be discussing how cells transport materials, and of course the cell membrane plays a huge role in that aspect. So in order to be able to study about cell transport, we need to have a really good understanding of the cell membrane. Remember, the cell membrane controls the movement of materials into and out of the cell. That's the definition of what the cell membrane is. It's important to note, however, that the cell membrane is selectively permeable. And what that means is that it only allows certain materials in and out. It doesn't just do this passively, though. It does it selectively. So it picks and chooses what it wants to come in and what it doesn't want to come in, to a certain extent, which is a good thing. Gives the cell control over what it brings in and what it releases. To understand a little bit more about how the cell membrane works, we're going to look at what the cell membrane is composed of or made up of. And the cell membrane is made up of a chemical called a phospholipid. This chemical contains a lipid and a phosphate group, and that's how it gets its phospholipid name. Confusing, right? Remember, lipids are actually fats and oils. Now here is a picture that you're going to draw of a phospholipid. It kind of looks like a sperm with two tails. So you're going to draw this and you're going to label it after you've taken your notes on it. And this particular phospholipid has two parts. All phospholipids really have two parts. They have hydrophilic heads and hydrophobic tails. Okay, so this is the lipid part, and this is the phosphate part. Okay, the circle and the tails. So make sure you draw that. Now, because of the two parts that it has, the hydrophilic, which means water-loving, and the hydrophobic, which means water-hating, the cell membrane actually arranges itself into what we know as a phospholipid bilayer, bi meaning two. So it's a double layer, double phospholipid layer. Now why does it do this? Well, because the hydrophilic ends like the water, so they put themselves close to where the water would be. Let's say that this is the outside of the cell and this is the inside. There'd be water out here, there'd be water in here. These guys like to be near the water, whereas these guys, the tails, the lipids, the fats, we know, don't like to mix with the water, right? Oil and water don't mix. Fat and water doesn't mix. So these don't like to mix with the water. So they orient themselves. It happens automatically. If we were to separate these, they'd go right back that way. What does this structure then have to do with anything that we're trying to talk about? Why is that? Why is the structure important? Why is it important that it has a bilayer? Or why is it important that there's a hydrophilic and a hydrophobic end? Well, let's talk about that. The phospholipid bilayer is very tightly packed together and only allows very small, they have to be small, non-polar, they can't have a charge, molecules through. For example, water, which is relatively nonpolar, can pass through the spaces of the bilayer. Na plus, or sodium ion, which has a charge, can't because of its charge. Okay, so in this picture we can see here's water, H2O, trying to get through. It goes no problem. And here's sodium, and it bounces right off. Okay, so in my picture here, I've drawn it. You need to draw this, by the way. In my picture here with my, I figured I'd draw because, my drawings are way better. As you can see, like this is way better than this. I don't even know why I used this. Anyway, so you can see water going through. Here the sodium bouncing right off. I particularly like the animation there. You see that? Bounce. I mean, it's just It's amazing, really, what I can do with these videos. Okay, but the key takeaway from this is that only small, relatively nonpolar molecules can go through the membrane, whereas anything big or polar or charged bounces right off. Because only certain materials can pass through the cell membrane has to design ways for other things to get in. And how it does that is it makes passageways through the membrane to allow other molecules in. I want you to draw this. Okay, so these little passageways, as you can see, are called channel proteins. They're proteins that are embedded in the phospholipid bilayer that allow specific molecules through and can be opened or closed depending on what the cell needs. So in this case, remember sodium before was charged, bounced off, couldn't get through. Well, if it gets to this gate, this channel, it can get in. Notice that this channel is specifically designed to fit the sodium, whereas this channel wouldn't let the sodium through because it's too narrow, but it does allow the glucose, which is too big to pass through the spaces, allows it to get into the cell. And that's important because the cell needs to be able to bring these materials in if it wants to. Obviously it doesn't want to all the time, but when it does want to, it needs to be able to bring them in. Okay, so channel proteins are very important parts of the cell membrane that allow bigger and charged molecules to get in when the cell needs them. Another feature of the cell membrane are the proteins that are located on the outside of the cell. These are called glycoproteins. Glyco standing for sugar and protein standing for protein. That's a shock. So sugar proteins. So it's a combination of the two different types of molecules. And as you can see, these are the glycoproteins here. If we pretend that this is the inside, this is the outside, these are glycoproteins. You can see them kind of squirrely little things, squiggly little things on the outside here. Now, they essentially, especially for Bio-11, they serve two functions. They act as cell markers. And what do cell markers do? Well, cell markers distinguish a cell, one cell from another. In our bodies, in particular, with our immune systems, they can distinguish our own cells from a foreign invader's cells. And that way our immune system knows which cells to attack and fight off. They also act as cell receptors, which collect messages from other cells. See these little areas here that looks like they're kind of like Y-shaped. It looks like something could fit in there. Those would be glycoproteins that accept messages. So the message would kind of stick right in there. And that would allow the cell to get a message from other cells in the body, which is kind of cool. Okay, so there are other types of proteins in here, as you can see. We talked about channel proteins. For Bio-11, channel proteins and glycoproteins are the only two you need to know. For AP Bio, you'll learn about all the different ones. But for Bio-11, the glycoproteins and the channel proteins are the two main ones you need to know. Finally, the model for the cell membrane that you'll see in the textbook is called the fluid mosaic model. And what that means, why we have this model of the cell membrane, is that it's made up of many different parts. Move that over there. Many different parts, and is also able to move in its liquid environment. So it's got many different parts and it's able to move in its liquid environment. And what I have here is a picture of a mosaic piece of art. Now, those of you that take art know that mosaic is essentially just a picture or something like that that's broken up into tiny pieces. Well, if you think of the cell membrane, it has all those different parts. Okay, but yet it's also fluid. It can move, it can change shape. And that in a nutshell are the basics behind the cell membrane. Make sure you bring your questions to class.